Hello everyone and welcome to today's webinar, Single Cell and Label-Free Binding Affinity Applications with Genentech. I'm Shijie Valdez of LabRoots and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Biosensing Instrument. To learn more, visit them at biosensingusa.com. Now, we encourage you to participate today by submitting any questions you might have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you are having any trouble seeing or hearing this presentation. I'd now like to welcome our speakers, Elaine Mai, Senior Scientific Researcher at Genentech, and Dr. Shijie Wu, Application Scientist at Biosensing Instrument. Before handing it over to our speakers, we have two questions for our audience members. Our first question, have you heard about SPR microscopy? And now our second question, what tool are you using to measure affinity and kinetics? And thank you very much for your time answering those questions and your participation. Dr. Wu, welcome. You may now begin your presentation, sir.
Thanks for the introduction. My name is Shijie Wu from Biosensing Instrument. Today I'm going to talk about the basic principles and applications of surface plasma resonance microscopy. Before we start talking about SPRM, please allow me to say a couple words about Biosensing Instrument. Biosensing Instrument is located in Arizona, USA, and we started in 2005. So since 2005, we have developed a series of platforms for specialty SPR measurement. Particularly in 2017, we developed this new platform called SPRM, which is very well recognized by the industry and received quite a few awards from different institutions. And it's also very well recognized, received a lot of interest from researchers all over the world. SPRM is designed for measuring the binding kinetic between molecules and membrane proteins. We all know that membrane proteins are the starting point for disease treatment. It actually accounts for over 60% of the drug targets today. So it's important for pharmaceutical researchers to understand the binding kinetics between the drug molecules and the targeted membrane proteins. On the other hand, SPR is a mature and widely used technique for measuring the binding kinetics between different molecules. However, to use SPR for measuring the binding kinetics between the drug molecules and the membrane proteins are difficult. This is because to use SPR, it usually requires the membrane proteins to be purified and immobilized onto a chip surface. The process of purifying those membrane proteins are usually extremely difficult and time-consuming. And also, those purified membrane proteins will lose their physiologic condition and therefore may affect their reactivity with the drug molecules. SPRM is designed to solve this problem. SPRM is a label-free technique that allows you to directly culture a cell onto the SPR chip surface. Therefore, you can study the binding kinetics with membrane proteins directly on the cell surface, close to their physiology conditions. It can provide a real-time analysis and give you the binding parameters, including the association and the dissociation rate constant and the binding affinity. It can also be used to study some nanostructures and nanoparticles, including species like virus, bacteria, and so on. SPRM has three key components. The first component is the SPR unit. It includes the light source, the detector, and the sensor chip. On top of this SPR unit, it is a bright field optical microscope, which essentially illuminates the same area, or looking at the same area. This optical microscope allows you to precisely position and locate the sample to where you want to do the experiment. It also allows you to monitor the status of the sample during the experiment. The last component is a uniquely designed flow system. And this uniquely designed flow system provides the optimized flow conditions to ensure accurate SPR measurement. It also allows you to directly culture the cells onto the chip surface and maintain the measurement conditions during the experiment. By combining all three of the components, and the system allows us to measure the parameters of the binding kinetics, including Ki-on and Ki-off, and the binding affinity. So how SPRM experiments are done exactly? Well, we know for conventional SPR, we use a focused laser beam and a uniform chip surface, and then get an average SPR signal for the whole chip surface. For SPRM, on the other hand, instead of using a focused laser beam, we use a collimated light source to illuminate a fairly large area of the chip surface. And then for each spot of the illuminated area, it will give one unique SPR signal. Now, if you map the SPR signal for each spot in the illuminated area against its XY position, and you will get a so-called SPRM image. Of course, if the surface is uniform, you will get a flat image with no contrast. Now, if the surface is not uniform, for example, if we have the cell on the chip surface, and then the cell will have different physical properties against its neighboring area, and which will in turn give different SPR signals. So the mapped SPRM image will reflect the position and the physical properties of the cell. If you introduce some chemicals into the system, and those chemicals start to bind with the cell on the chip surface, and this binding will affect the physical properties of the cell, such as the stiffness and the surface morphology. And this change will in turn affect the SPR signal and can be recorded in the SPRM image. And the whole binding process, the association and the dissociation, can be recorded using a series of SPRM images. Now, once you have recorded the whole process, and you can go back and extract the information for any particular spot in the recorded image, and then plot it as a function of the recording time. And then you will get the so-called SPR Sensor Graph. And this SPR Sensor Graph will carry all the information of the binding, the association, and the dissociation. And then you can analyze the signal against the theoretical models, and from that you can extract the binding parameters, including the Ki-on and Ki-off, and the binding affinity. Well, the typical step of SPRM experiment includes the following. First, you will prepare the chip surface, and then you culture the cell onto the chip surface, and then you supply the running buffer. And when the system stabilizes, you will get a stable baseline for the system, and then you introduce the molecules. The molecules start to binding, you look at the association, and then you cut off the supply of the molecules and to look at the dissociation. And then you repeat this whole process for many different concentrations of the molecules, and you obtain the so-called profile of kinetic titration type of sensor graph. And by analyzing the whole data series, and you obtain the kinetic parameters of the Ki-on and Ki-off, and also the binding affinity. And on top of that, you also get a histogram which reflects the statistical distribution of those parameters across the whole studied cells. To compare the difference between the SPRM and a conventional SPR, so the conventional SPR uses a uniform surface, and the protein has to be purified and immobilized uniformly onto the chip surface. And then you measure an averaged single signal for the whole surface. For SPRM, on the other hand, you don't have to purify the protein. You actually culture the cell or grow the cell directly onto the chip surface. And because you can have many cells onto the chip surface, so the surface is not uniform, it's heterogeneous. And then you can actually measure the responses from all the individual cells, and which give you a localized variant. So that's why SPRM not only can give you the binding kinetics, the binding information for the average cells, but also can see the difference between different cells. So you can see the heterogeneity between different cells or even different parts of the cell. So the major advantage for SPRM is you don't have to purify the protein. You have a physiology condition close to the native condition of the membranes on the cell. So this is one of the earliest experiments we have done using SPRM. So the system what we are looking at is the binding affinity of glycoproteins on single cells. So we introduce WEA to the system to bind with the sugar residues on the surface. So the optical image of a single cell is shown in this image, and the corresponding SPR image is shown in here. And if you look at the two images, the perimeter of the optical image looks smaller compared to the SPR image. This is because the optical image doesn't have the resolution to see the spread, the very edge of the spread cell. Therefore, the SPR image gave you a more complete and with more details of the cell. The binding process is shown in this movie here. So first of all, you have a baseline, and then you introduce the WEA, it starts binding. The intensities of the binding site start to increase, and then you start to dissociate, and the intensity will decrease accordingly. And the jump in this place corresponding to a process we call the regeneration, which we introduce a chemical to remove the WEA completely from the cell surface. And after that, the baseline essentially go back to the same level as before the experiment starts. So the whole process is recorded in a series of images, as I said before. So for example, this is one of the images recorded at 550 seconds, and which is about here, so close to the end of the dissociation process. And with all the images recorded, we can analyze all the data at different locations, and then we get a map which reflects the variation, the localized variation of the binding activities across the cell surface. So the map images shown here are the K-on and K-off of the WEA binding with the receptor with the proteins on the cell surface. Now I will show you a number of examples for the binding measurement using SPRM. So this is one of the nice experiments we've done with AstraZeneca. And the system we are looking at is a small molecule, AZ1395, targeting the GPR39 receptor on HEK cell. The result is shown here on the left. This is SPR image, and on the right is a corresponding optical image. And the optical image, you can see more clearly where all the HEK cells are located on the chip surface. And each of the color square on the surface reflects one of the good areas, give you a good binding data. And you can see immediately the good binding data overlaps with the position of the cell very well. So this tells us we are looking at the bindings happening on the cell, not on some empty areas. And with all the data we analyze, we can obtain the isotherm from which we can obtain the binding affinity based on the thermodynamic analysis. We also have all the sensorgrams for each of the unit area, and we can feed the kinetic model, the one-to-one binding kinetic model for each of the sensorgram, and from the fitting, we can obtain the value, the average value of the K on and K off, and also the binding affinity. And also we see the, because we are looking at many different areas, and we get a distribution histogram, which shows the variation or the heterogeneity between the different cells or the different parts of the surface. So the results we obtained for the binding affinity is about 400 nanomolar, and the corresponding K on and K off reported also here. And this molecule is actually very well studied by AstraZeneca and other researchers. So one of the results reported in the literature using a technique called the BSI, which is the backscattering interferometer, and it gives a value about 233 nanomolar, which agrees very well with our measurement down here. This is another interesting experiment done by Signature. The system we're looking at is a small molecule targeting the anti-S1 receptor, which is endogenously expressed on breast cancer cell. Again, the results is shown on the top images. The middle image, which is the optical image, and you can see where all the cells is located. And the corresponding SPR image is shown on the right. The left side is a fluorescence image, and used to validate or to prove the expression level of the anti-S1 receptor on the cell. The analyzed data is reflected by those colored squares on the image, and again, it actually overlaps with the position of the cells very well. And the results we obtained from the measurement and gives affinity, the big Chi-D value, about 2 nanomolars. This particular receptor, the anti-S1 receptor, this protein, actually has been purified and studied by Dr. Huber from Roche in Britain, UK. And she actually used conventional SPR and purified this receptor and did a conventional measurement for the same molecule. And the results she reported is about 400 picomolars for the binding affinity. In the literature, there's also some in vitro studies for different type of anti-S1 receptors. And for the wild type and human type, it gives different values, about like 500 picomolars and to 8 nanomolars. And our SPRM gives data about 2 nanomolars, so which actually fairly agrees very well with those in vitro studies. This is one of the experiments done by Daiichi Sankyo in Japan, and which is looking at the binding of histamine and the GPCR receptors on HEK cell. On the left side, those are the positive binding reactions of the system, and on the left is the optical image, which shows all the HEK cells on the chip surface, and the on the right, this is the SPR response image, which shows actually the binding activity occurring, which is actually maps very well with the location of the cell, first of all. And from the data, we can analyze and obtain the binding affinity to be about 7 nanomolars between the histamine and the receptor. And this is experiment is actually being verified using a negative control. For the negative control, we actually use the same cell. On the left, that's the optical image. And on the right is the SPR response. The difference here is we use the mutant histamine, which shouldn't show any binding activity. And indeed, from the SPR response images, we see very low levels of signal, which shows there's no or very small binding levels between the molecules. Here are three more binding measurements using SPRM. So the first one on the left is done by Takeda, and this is a small molecule targeting the GPCR receptor on the cell surface, and we see very positive binding activity on the cell, and then we have this very nice distribution curve, and get an average value for the binding affinity is about 8 micromolars in this case. And there's also very interesting questions regarding the ion channels. So this is an example showing the binding of a small molecule to the protein that regulates the ion channels. And again, the optical image and the SPR response image, and we see the very nice binding responses of the cells. And then the results we get for the binding affinity is about 2.6 micromolars in this case. And the last example shown here is actually an example done by a biotech company in San Francisco. And we looking at here is a small molecule binding to some receptors on the HEK cell. And again, the top one is the positive measurement. And you can see this very nice positive reactions with a value of 865 nanomolars for the binding affinity. And this measurement is checked against some negative controls. So actually two different types of negative controls was performed. The first one is a negative control compound, which means it's a positive cell, but a compound that will not bind to the receptors on the cell. The second one is a negative cell chip, which is the HEK as the cell which is not overexpressed with the receptor. And both of these negative controls has shown very low binding activity, so which further verifies the result we obtained on the positive cell. So far, I have shown you a number of examples for the binding between small molecules and the membrane proteins. And here is actually an antibody targeting the membrane proteins, which is actually a membrane transporter in this measurement. Again, the results is shown here. We have see the very nice good binding sites overlap with the position of the cell, and the good results from the analysis and gave the nice distribution and the average values for the binding affinity and the K on and K off. The binding affinity for this particular reaction is about 2.9 nanomolars, which agrees very well with the expectation from the customer. So all the examples I showed earlier are done with cells that are fixed onto the chip surface. So one of the common questions people are asking is, can we do live cells, particularly with those people who are studying antibodies? So this experiment is a good answer to this question. So what we show here is a full antibody binding to some live cells in the growth medium. So if you look at the image of the system, so on the left, this is actually the running buffer, shows a color red. And if you look at the syringes, even here, it's also a little bit red color. So which tells we are using the growth media in this case. Again, the measurement result is fantastic, and we see the optical image and the corresponding SPR image, and also the good binding active area overlaps very well with the position of the cell. And from the data, we get this nice distribution curves and the average value for the binding affinity of this antibody to the cell. It's about 1 nanomolar in this case. Because SPR measurement requires the cells to stick to the chip surface without any movement during the experiment, so it's easier to work with the adherent cells instead of suspicion cells. So the question is, can we actually measure suspicion cells at all? The answer is yes, thanks to one of the researchers and customer from Pfizer. He is working with B cells, and he developed the protocol to grab the B cell from the solution and stick it onto the chip surface, and then do live measurement with the antibodies. So this experiment shown here is the proof concept. And the analyzer used here is the WDA. With all the nice data sets shown here, it proves the system is stable during the whole measurement. And of course, Pfizer didn't stop with the WDA, and they move on with this protocol, with this procedure developed to study the real bindings between antibodies and the receptors on B cells. Besides measuring the binding kinetics with membrane proteins, SPRM can actually have many other applications. For example, it can be used to visualize the position and the movement of nanoparticles on the chip surface. We know the surface plasma wave travels along the interface of the chip surface. When you have a nanoparticle on the surface, it will change the local electron density, which in turn will interfere with the travel of the plasma wave. The result is you will see a unique scatter pattern on the SPRM image. And this unique pattern allows us to recognize the existence or the presence of the nanoparticles on the chip surface. Plus, the intensity of this unique pattern is actually directly proportional or related to the size and the distance of the nanoparticle from the chip surface. This experiment shows the relationship between the intensity of the image and the position of the nanoparticle. The nanoparticle is charged, and we apply an oscillating electric field, and then the particle will oscillate up and down at the close to the chip surface, and then the intensity will oscillate accordingly. And in this image here, it shows the distance and the intensity of the interference pattern. One of the interesting applications based on the activities of nanoparticles is to look at the effect of antibiotic on E. coli bacterial cell. So the bacterial cell is sitting on the chip surface and it's jumping up and down when it's alive. So once you introduce the antibiotics to the solution, and it will kill the bacterial cell, and then it will lose the movement. So the experimental results shown here. So the images in the when the cell is alive, and you can see the position, and based on the intensity of the image, and you can see it's jumping up and down at the surface. And once you introduce the antibiotics, and it gets killed, it starts to be a constant position, not moving anymore. And this results is actually shown in this movie here. So when the cell is alive and when it's become dead, killed by the antibiotics. SPRM can be combined with other techniques like electrochemistry. So we can actually add a potential stat to the system, and then we can supply electric modulation to the system, and then using the SPR imaging capability to monitor the local responses of the chip surface, the materials on the chip surface, to the electric modulation. Here are two examples for the electrochemical applications with SPRM. The first one is the SPR-based impedance experiment. So what you're looking at is the responses of SEHA cell to the apoptosis treatment. And you have the optical image, the SPR image, and the SPR-based impedance image. So the SPR-based impedance image shows the localized responses of those cells to the treatment. And the second example, it shows the CV, using the CV to study the individual gold nanowires on the chip surface. So each of the gold nanowire was shown up with the interference pattern on the image, and they can be labeled from here. So for each of the gold nanowires, you can have a CV based on the SPR signal. So you can see depending on the size and the conductivities and those different properties of the individual gold wires, and they should see differences in the CV. So which proves those nanowires are not uniform. They have different physical shapes and scales and properties. SPRM has wide applications. So here is a short list of some selected publications using SPRM. In summary, SPRM is a label-free technique that allows you to measure the binding kinetics with membrane proteins directly on cell surface. And it provides a real-time analysis for binding kinetics and binding affinity. And it can look at single cells and also can look at the heterogeneity between different cells. It can also be used to study the nanostructures and nanomaterials. You're welcome to contact Biosensing Instrument for any questions or interests. And with this, I will pass the time to Mrs. Elaine Mai for her interesting research. Thank you very much.
Thank you, Shijie. My name is Elaine Mai from Genentech. My talk today will be about evaluating Biosensing Instrument's SPRM200, which can differentiate between mono and bivalent binding of anti-HER2 antibodies to high and low HER2 expressing cells. Before I delve into the evaluation, I will give you some background. In the context of antibody development, it's very important to know its affinity and kinetics parameters, since antibody binding to its antigen plays a key role for antibody selection. As most of you may know, K-on is the association rate constant and K-off is the dissociation rate constant. Currently, antibody selection relies heavily on solid-phase biophysical methods such as SPR and octet. Here is a cartoon of how SPR works. SPR basically measures the refractive index. The antigen or antibody is immobilized on the chip, and then the ligand flows over the chip and the binding event is recorded. This method is amenable for screening antibody drug candidates, label-free and very robust, but it has its challenges. And some challenges to determine affinity are that some antigens cannot be expressed as soluble proteins, especially for multi-transmembrane receptors. The technique used for immobilization may impact the affinity measurement as well. But most of all, purified proteins do not reflect the native structure, function, and expression of the multi-transmembrane proteins. And this is where determining antibody affinity and avidity on cells becomes very critical. And especially with the advent of new antibody modalities, as shown right here, these past years with more and more complex formats with multiple arms that play on avidity. I will show an example to demonstrate why it's very important to understand the cell binding affinity. In this example, TDBs, as shown here on the left bottom, are T-cell dependent bispecific antibodies with two arms, one with affinity to the T cells and another to the tumor cells. One of the main challenges is to prevent on-target, off-tumor binding to healthy tissues. This extensive work that I will be discussing afterwards demonstrated the fine-tuning of affinities of each arm is critical for efficacy and tolerability. Here is the summary of the great work performed by the AntiHer2 CD3 version 2 TDB team led by Timo Juntila, where they engineered different antibody formats that would favor binding to tumor cells expressing high HER2 and protect the healthy tissues that may express low HER2. For that, they either have the IgG TDB or 1-Fab IgG TDB format. The 1-Fab IgG TDB is engineered to have various low to high affinities to HER2. When they compare the binding of all those molecules on either SKBR3 that express high HER2 levels or MCF7 that express low HER2 levels, there was no difference in binding to SKBR3 for all the molecules. However, when binding to low HER2 expressing MCF7 cells, these antibodies show a big difference in binding. I want to point to the lowest affinity antibody format that showed almost no binding here on the right B plot in red. And this is correlated with a cell killing experiment where they looked at killing on high versus low expressing cells. They saw strong killing on high expressing cells for both formats and no killing on the low expressing cells for only the 1-Fab IgG lower affinity format, demonstrating they can increase the therapeutic index by playing on avidity. This also demonstrates that avidity can rescue affinity while preserving antibody specificity. And as such, you can imagine how understanding avidity can play a critical role in antibody selection. In summary, antibody cell binding affinities matters a lot for informing tissue distribution, target engagement, PTPK modeling to inform dosage requirement, differentiating antibody binding sites, and lead identification when antigen cannot be expressed as a soluble protein. And as such, with an ever-increasing need to study binding of new antibody modalities with their relevant antigen on the cell surface, we evaluated Biosensing Instrument's SPRM200 as a potential platform to study affinity and avidity. Hence, we sought to design an experiment to determine if we can differentiate between monovalent and bivalent binding to various cell lines, such as SKBR3 and MCF7, with different expression levels. The SKBR3, as mentioned, expressed high HER2 levels, and MCF7 line expressed low levels. We started with fixing the cells with 4% PFA in-house, and then we shipped the chips with fixed cells, antibodies, and buffers to BI for measurement. The antibodies tested are an isotype-controlled anti-GD human IgG, one-arm TDB antibody with monovalent binding to HER2, and Herceptin with bivalent binding to HER2. Here are the results that we have. And as you can see, for anti-GD human IgG isotype control binding on SKBR3 cells, it shows that there was no binding as expected. And also, there is no reliable binding to determine affinity and kinetics in this case. Here are the results we have with the specific antibodies. On the high expressing line SKBR3, the 1-arm monovalent TDB antibodies show only one histogram for binding events, or ROIs. Only one histogram that allowed determination of 1 KD. Whereas Herceptin, the bivalent antibody on the bottom, shows 2 unique histograms of ROIs. Note the total number of ROIs are almost equivalent, 58 versus 66, for both the low and high affinity interactions with the bivalent antibody. I will compare this to MCF data later. With the lower HER2 expressing MCF7 cells, again, the monovalent TDB antibody shows only one histogram for binding events or ROIs, whereas Herceptin, the bivalent antibody, shows 2 unique histograms of ROIs, as expected. And as you can see, the high affinity interaction, which is around 168 picomolar on the low expressing MCF line, relative to the lower affinity interaction of about 3.3 nanomolar of Herceptin, has almost a twofold difference in ROI, 45 versus 81. And notice the lower number of RIs for the higher affinity sites. And most of the RIs for bivalent binding are lower affinity for the lower expressing line. Here are the results we have comparing the two cell lines with Herceptin side by side to demonstrate that the binding density as shown by the total number of RIs show a difference between the two lines, which indicates there is an avidity effect. Specifically in the high expressing line SKBR3, there are more binding events with higher affinity than the MCF7 line, pointing to a higher avidity effect on the high expressing cells. This extra level of information on receptor binding is unique with SPRM for affinity and avidity determination, and thus can be a very useful tool for studying multimeric molecules at Genentech. In summary, we have compared the affinity with other technologies as well internally, and I am just showing you one of them today. And here, as you can see, SPRM's affinities are very similar to Kinexa's, and also the K-on and K-off are in line with what we expected. The main advantage of SPRM 200 over other assays, such as FACS, is that there are very few technologies out there that can determine kinetics and avidity on cells. Since there is a true need to determine binding kinetics, SPRM is a promising technology due to its ability to detect multiple association levels with multivalent molecules using their spatial mapping function. We look forward to testing the instrument ourselves in our lab. There are also lessons we learned from this technology. Overall, there was reliable generation of affinity and kinetics parameters for SKBR3 and affinity for MCF7. However, the technology is not suitable for cell lines expressing less than 10,000 receptors of interest. And as you can see in the MCF7 line, there was no kinetics parameters generated. Last but not least, I want to acknowledge the successful work that has been done here involves great collaborations across different teams in-house and externally. Specifically in-house in our department, the Biochemical and Cellular Pharmacology group is DeBarco and Leticia, who leads the team, and Mercedes, Yichin, and Margaret, who is our BLT members, and also the internal antibody engineering group, Rajesh and JT, for their support and also sharing of information. And externally, I would like to thank Elizabeth from Sapadyne and Shijie and Akimi from Biosensing Instruments for the evaluation of this instrument. And this concludes my talk, and thanks for your attention.
Now we're going
to start the live Q&A portion of the webinar.
Thank you, Shijie and Elaine, for that informative presentation. We will now start the live Q&A portion of the webinar. And if you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's take a look. We already have some great questions coming in from our audience member. This first question is for you, Shijie. Surface of this cell has many receptors. How does SPRM detect the binding of my specific target receptor?
Thank you. Yeah, I mean, this is actually one of the common questions people ask, and it's also actually an important steps for experimental design. So there's two ways or two procedure, two experiment steps we use to guarantee that we are marrying the right receptor. So first of all, if you have different receptors, so we need to overexpress to really express the targeted receptor on the cell. And the other things is also in the experiment we include some negative samples, either a cell with a negative cell with no expression receptors, or negative chemicals, just to check we are measuring the receptors.
Thank you so much, Shijie. And our next question is for you, Elaine. What was the minimal HER2 copy number required to see cell killing with the 1-Fab IgG TDB molecule?
That's a good question. So none of the cells that express less than 200,000 HER2 per cell were sensitive to 1-Fab IgG TDB, whereas all cell lines with greater than 200,000 HER2 copy number were readily killed by the 1-Fab IgG TDB.
Thank you so much. And Elaine and Shijie, our next audience member wants to know, what are the requirements for receptor levels in cells to detect the binding event, and can it measure an indigenous cells?
So what I've shown in my slide, we can't do measurements below 10,000 receptors per cell.
Yeah, I mean, basically, we are trying to look at a particular receptor on the cell, and then the good thing will be if you have extensive amount of those receptors, so you have a stronger signal. So the bottom line is the more receptors on the cell, and the better. And then we're really looking, targeting, like a 25K or something, and or higher. And the thing is, it's difficult to really know the exact number of receptors. And so if you can overexpress it, the more the better.
Thank you so much, Shijie. And I want to thank our audience for these great questions coming in. We have time for a few more. Shijie, what is the range of affinities that can be measured by SPRM?
Well, the instrument is actually sensitive. It's reliably measuring from picomolar up to below millimolar range.
Thank you. And what about the range of affinities that can be measured by SPRM?
That's basically the question I just said. Is in the range of picomolar affinity up to below millimolar affinity range.
Thank you. And Shijie, could you explain a little bit more about why SPRMs show two peaks in the histogram?
This is an interesting question. Actually, Elaine just showed the results in her presentation. So the synthesis is based on the SPRM. So the SPRM is not like the conventional SPR. So it's we call it imaging technique. So you basically are looking at a big area, and then you divide the whole area into small regions. So each of the regions, it can give as a measuring unit. It can give you one sensor graph, one SPR response. So this small region can cover actually maybe part of the cell, which have receptors, located in this region. Now, so if you have like a different type of receptors or different binding molecules, so bottom line, so if you have different reaction kinetics responses, and then this SPRM will look at those responses locally, and at the end, so you get collect all the responses from all over the surface, and you put them together as a histogram, so you get a statistically global picture of the responses from the whole cells and the whole samples. have those different responses, and you will be able to see like, 2 peaks or multiple peaks even, and reflect the fact that you don't have the same type of, you have multiple different type of binding responses on the surface.
Thank you so much. We have time for one more question. How do you capture the suspension cells on the chip, Shijie?
Yeah, I mean, the SPRM requires, the cell, it's like an SPR experiment, so the cells cannot be moving during the measurement because, they need to be stable there and stay there. So for achieving cell is easier, and for the suspension cells, they flow in the solution, and usually we can capture them with, some chemicals coatings on the surface. And the example I show you in the slides is from Pfizer, and they use the cell tag, but different chemistries can be used. For example, like polylysine, PFLD, those. There are different protocols you can use to capture those suspension cells from the solution and hold them onto the surface and do the experiment.
Shijie and Elaine, I want to thank you so much for your time today and for your important research and for your presentation. I'd also like to thank LabRoots and our sponsor, Biosensing Instrument, for underwriting today's educational webcast. And before we go, I want to thank our audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and bye-bye.